Hi everybody, my name's Porik, and this will be my mini presentation on the CRISPR-Cas9 mechanism. So it's important to remember that there's actually three types of CRISPR systems, and they differ in their usage of Cas proteins and molecular pathways. However, all types use Cas1 and Cas2 proteins. And the type that's relevant to gene editing is type 2, and this uses a Cas9 protein. So in this presentation, we will be focusing on the type 2 system. So how exactly does CRISPR work? We begin by having a phage inject its own DNA sequence into a bacterium. Cas9 is produced along with tracer RNA, and these two combine to form a complex, used to form mature CRRNA. CRRNA functions as a guide to the invading gene into the Cas9 system, where it will be cleaved and degraded. The cleaving ends are upstream of a very important sequence called the PAM sequence, also known as the protospacer adjacent motif, and this will tell us exactly where the Cas9 protein will be aligning. Here you see a visual of the PAM sequence relative to our CRISPR loci. Generally, the PAM sequence has a code of NGG. So let's begin with our animations. So first, we see a phage inject its genetic code into a bacterium. And we see here that this genetic code actually aligns with a CRISPR locus. There's one more thing that I'd like to add to our operon, a very important sequence that codes for the tracer RNAs themselves. When we do produce those tracer RNAs and the Cas proteins, the tracer RNA will align with the locus associated with the viral gene, and so will Cas9. This will form a complex to produce mature CRRNA. What we see here is the Cas9 protein has formed its complex along with the tracer RNA and the CRRNA. The CRRNA is base pairing with the viral DNA and will be pulling it through the Cas9 system, aligning it with those scissor-like enzymatic regions. Eventually, the target gene will be cleaved and degraded. So you must be wondering what the difference between this process and the gene editing process is. You'll recall the tracer RNA and the CRISPR RNA. These are going to be replaced with a sequence called singular guide RNA. Another question you might ask is, what happens after we remove the gene? We still have a double-stranded break. This can be repaired with two pathways, non-homologous end-joining repair and homologous directed repair. First up, we have NHEJ. This is extremely fast. It creates indels, which is a collective term for insertions and deletions, and overall it inactivates a gene. In order to signal that there's a double-stranded break, we have a Ku heterodimer align over the double-stranded break, and these recruit two DNA kinases along with DNA ligase to ligate the two strands together. This is definitely not the best for creating a working gene. That's why we have the homologous directed repair pathway. Though it's not as efficient as NHEJ, it will result in a working gene. The first step is two mRN complexes recognize a double-stranded break and signal for endonucleases to eat away at the ends to expose 3' bases. Here we see two holiday junctions are formed with our desired gene and 3' bases that are exposed. This allows DNA polymerase to move across and add complementary sequences of the desired gene onto the broken strand. The new bases are ligated. Resolvase will have undone our holiday junctions, resulting in a singular completed strand. The other broken strand will have complementary bases added using DNA polymerase, giving us a corrected and working gene.